Hello everyone, my name is Alexandru Sasuclark, a University of Hawaii PhD candidate in the Cell and Molecular Biology Department and Toby Lee ARCS Award recipient. Today, I would like to talk briefly about my research, the cells I study, and the impact this may have on human health. The research I am conducting alongside my mentor, Dr. Matthew Pitts, involves studying a GABAergic interneuron, known as a Parvalbumin interneuron, and an extracellular matrix structure that selectively surrounds Parvalbumin interneurons, known as a perineuronal net. In the brain, GABAergic interneurons act to suppress the firing of other neurons. Parvalbumin interneurons in particular exact fine control over neural networks, suppressing many neurons. This function is critical because it prevents aberrant signaling between neurons. Fine control requires that Parvalbumin interneurons interact with multiple neurons, and more critically, the correct neurons in a network. Perineuronal nets, as the name suggests, are an extracellular matrix-derived lattice-like structure. These structures help to stabilize synaptic connections to the Parvalbumin interneuron, allowing it to function optimally. These nets have an additional function as well. They protect the neuron from reactive oxidative species. Due to the regulatory function, Parvalbumin interneurons are highly metabolically active. Utilizing a significant amount of energy requires that they have many mitochondria, the downside being that they produce many free radicals. The nets help protect these neurons from some of this stress, but one final piece is needed for the cell's survival in this environment. The selenoprotein, characterized by the incorporation of selenium in the form of selenocysteine, is crucial to the cell's survival. Most selenoproteins are redox enzymes, and Parvalbumin interneurons require these proteins to reduce the effects of the free radicals they produce. So what do my mentor and I study exactly? Well, we are interested in how selenium impacts the development of the Parvalbumin interneuron and the perineuronal net. To do this, we collect neurons derived from embryonic mice and assess their development in a primary neuron glia co-culture model in various concentrations of sodium selenite. Controlling the selenium environment allows us to challenge our neurons by giving them too much, too little, or just enough selenium to produce the selenoproteins they need. Finally, controlling the interaction between the glia and the neurons, including what glia and neurons get to interact, we can determine what selenoproteins are predominantly produced to assess the functions of those selenoproteins on our neurons' development. To end, I would like to touch on why this matters to us. In the brains of those with neuropsychiatric disorders, such as schizophrenia, Parvalbumin interneurons and perineuronal nets are noticeably affected, with far fewer of these neurons and nets reaching maturity. Because our research focuses on determining the developmental role of selenium on Parvalbumin interneurons and perineuronal nets, coupled with other methods, it may allow for better accuracy in determining an individual's outcomes of developing certain neuropsychiatric diseases. I would like to thank those that nominated me for this prestigious award, and the judges watching my presentation. Wherever you may be, have a wonderful evening or day, and thank you for the opportunity to share my research with you.